today we are going to talk about a few of the applications of eukaryotic gene regulation. So these are some of the more obvious things that we can see that are affected by how eukaryotic cells regulate their genes. When we talk about eukaryotic gene regulation today, we're going to look primarily at humans or animals. Remember, eukaryotes have a complex system of gene regulation, of enhancers and such, that aren't really related to the activator or the operon of prokaryotes. Alright, so the first thing we're going to look at is what's called RNAs and non-coding RNAs. So sometimes we represent non-coding RNAs by making little nc and RNA. Those are non-coding. And what I mean by non-coding is that there's a lot of DNA that doesn't make, is transcribed into RNA, but doesn't make proteins. It's just, end goal is to make RNA that does different jobs. Even though RNA can make up ribosomes, mRNA that's translated into proteins, tRNA, others have different jobs. And one of those jobs is to regulate genes. And two specific examples of non-coding RNA are miRNAs, mi standing for microRNAs, and siRNAs, which are small interfering RNAs. And they both go into regulating gene expression after transcription has happened. So after DNA has been transcribed into an mRNA transcript, miRNAs and siRNAs help to regulate what happens to that transcript. Does it get translated or not? So some of them can prevent a mRNA from being translated into a protein by binding to that mRNA. So it's kind of a key. It's seeking out certain mRNAs that are a complement of these silencing RNAs, and these silencing RNAs are like 7 to 10 to 15 around there, base pairs long, and it binds to the complementary mRNA, and it causes it to disintegrate. So, for example, if here maybe is my mRNA molecule, my let's say maybe siRNA molecule would be up here, and it seeks for a complementary mRNA, and when it binds to it, it causes it to disintegrate, to be disintegrated and to dissolve. And so it can block gene expression that way, and that's called RNA interference, or it can stop that mRNA from being translated, so it can just bind to it and stick on there and keep it from being translated. Another thing they can do is take part in what's called chromatin remodeling. And we talked about euchromatin and heterochromatin before, and euchromatin is very loosely packed DNA. And since it's loosely packed, RNA polymerase can get into it and transcribe a protein. And what chromatin remodeling is turning euchromatin into heterochromatin, which is very densely packed, and so densely packed together that polymerase can't get in there and translate it. And so some RNAs regulate gene expression by turning some euchromatin into heterochromatin to prevent gene expression, or turning some heterochromatin into euchromatin to prevent gene expression. So these are all roles that RNAs or non-coding RNAs play in help regulating gene expression. Alright. It's kind of the second part of here that we looked at in chapter 16. Really briefly, we're going to go over cell differentiation. So how differential expression of genes can determine what makes a liver cell from a brain cell, and how that's related to embryonic development. So when embryo is first dividing, how it can differentiate all its cells into all the cells of a human body. So every organism is made up of hundreds of different types of cells, and each of them are specialized. And the difference between these separate types of cells, like we talked about last time, isn't that they have different DNA, but that they express different parts of their genetic material. And that's fine when we're talking about adult organisms, but we all start out as a small little embryo. And all the cells in that first embryo are exactly the same. How do those first 16 cells or 8 cells turn into all the hundreds of different types of cells that we have in our adult body? Well, it all has to do with regulating the expression of these different genes. So, one part, egg cells, to start out with, already have regulatory information from their mother. So, egg cells, to start out with here, have information from their mother that determines what they're supposed to do next. So what parts of them are supposed to form the heart or the gastrointestinal tract, the notochord, those such things. And when a sperm cell arrives and fertilizes the egg, it drops off some of its regulatory genes called cytoplasmic determinants. And these things help to tell the embryo or the egg cell, the fertilized egg, the zygote, to start dividing into 2 cells. And depending on how they divide, different concentrations of different determinants will be in each cell. And those concentrations will tell each cell to furtherly divide and to do some separate things. In addition, embryonic cells also communicate with each other. So there's a bunch of different embryonic cells, and based on their position and location next to each other, they can signal each other through signal transduction. So here in this picture, signal molecules from one embryonic cell are telling a receptor on another cell to send a signal to the nucleus to divide more, or to divide in a certain way, or express certain proteins. And that way, the early embryonic cells can use positional information to help them differentiate and specialize. So the inside cells on the inside of this zygote, or this blastocyst, start turning into the gastrointestinal tract, and the outside turn into the epithelium and things like that. Certain other cells or genes, like in muscles, are called master regulatory genes. So, in different cell types, we have different master regulatory genes, and when they're turned on in certain cell types, they tell that cell to produce other proteins to help them be, for example, a muscle cell. So, for example, in a muscle cell, there's a gene called MyoD, which is a master regulatory gene for a whole host of other specific muscle-related genes. So, in precursor muscle cells, MyoD is turned on, and it codes for transcription factors that code for maybe other muscle fiber parts. It regulates the expression of other transcription factors to transcribe myosin or other muscle proteins, and itself. So, master regulatory genes that are activated in early embryonic development can help cause cell differentiation. And kind of lastly, there's homeotic, or what's called Hox genes. And what these are genes in the early embryo that are laid out in a linear map that matches the body. So, for example, on chromosome 3, the first or farthest to the 3' end gene has to do with what goes on in the development of the head. And they're kind of color-coded here along this Drosophila, this fruit fly. And the farthest towards the 3 end has to do with the anterior end of the fly, and the posterior, close to the 5 end genes, have to do with the abdominal portion of the fruit fly. Same thing happens in mouse and humans. These are highly conserved, ancient, ancestral gene sequences that help specific areas of the body differentiate into certain cell types, and they're laid out in that same topographic sequence on a chromosome. And so Hox genes are very ancient, preserved gene sequences that map anterior and anterolateral sections of the body in their development, and they're common amongst wide ranges of geni and species, and can be found throughout the life forms that we know of on Earth. So, a big thing in cell differentiation now is cloning, or reprogramming cells to differentiate where we need them to. So many of you have heard of cloning as far as Dolly goes, and I'm not going to beat that to death, but cloning in the terms of reproductive cloning means to make an exact genetic copy. So making an exact genetic copy is called reproductive cloning. And reproductive cloning has fallen out of favor, pretty much besides all of the ethical rationale, because we have more efficient techniques to do the same thing these days. And one of those things is stem cells. We can create stem cells that are capable of a wide variety of giving rise, to a wide variety of different cells, differentiating. So things like plants, all the adult cells in plants are what we call totipotent. Totally potent. What that means is if I cut off a piece of a leaf or a stem or a root, they're capable in the adult form of differentiating into any type of leaf tissue. That's why you can cut a potato in half and plant it again and it grows a new potato. Well, animal cells aren't the same. If I take a liver cell and I create a colony on an agar plate, for example, it will only give rise to more liver cells. It won't create brain and skin cells. It's not totipotent. It's specialized already. But we can create certain cells, or they're called stem cells, that are capable of differentiating into different types of cells besides just their own. Some of the key sources of stem cells are, of course, the embryo and in the spinal cord. And to clone an animal, what we do is take an egg cell, we suck out the nucleus of that egg cell and implant the nucleus of the animal we want to clone. We let it multiply and put it back in the uterus of a host organism. When it gives birth to a cloned animal. Creating stem cells is a lot easier. We were just creating cells that are, instead of totipotent, pluripotent. We can create stem cells that are capable of differentiating into many, many, many different types of cells, but not all, not totipotent. What we do in that case, kind of going back here, is we take this nucleus and we grow it into an embryo of undifferentiated pluripotent stem cells, and then we arrest the development there. We harvest these pluripotent stem cells that are capable of differentiating into many different types of cells. So we let our stem cells divide, and they divide into more stem cells, or pluripotent cells that can turn into fat cells or bone cells or white blood cells or anything. And then what we can do is we can take these precursor cells, and say you got a bad liver, we can coat that liver in these stem cells, and they'll all differentiate by given the signals for the liver cells next to it, into brand new liver cells. So we can culture embryonic stem cells, and we can put them in different cultural conditions, and they can differentiate into any stem cells that we want. We use this, for example, for burn patients. It's called a burn gun. You put a bunch of stem cells in basically a big painter's gun, spray it on a burn, and they differentiate into brand new epithelial skin cells to cover that burn. Or we can grow new organs by using stem cells. And using their pluripotence and using the fact that they haven't specialized yet. But stem cells are the only things that can do that, because our adult cells, if we cultivate them, will only produce more of whatever type of cell our original cell was. Pluripotent stem cells can turn into many different types. The last thing we're going to talk about gene regulation and differentiation is cancer. And this is going to kind of loop back to the cell cycle. There we go. And cell cycle regulation. So research has discovered cancer-causing genes. There's genes that, when things are mutated in them, cause cancer. One example are proto-oncogenes. So what proto-oncogenes do normally is stimulate normal cell growth. Tell your cell when to divide and that they should start growing because they need to. Running out of space, they have a lot of energy, they can do that. But when mutations happen, by translocation or duplication or point mutation, they may not function normally and cause rapid cell division or keep stimulating cell division and cause cancer. And when these mutations happen, they turn into oncogenes. So here we have a picture of 3 ways proto-oncogenes can turn into oncogenes: by translocation. So the gene, maybe this gene moves to a new locus that's really close to a promoter, so it gets transcribed a lot and it makes a ton of normal growth stimulating protein in excess. It can be maybe duplicated. So now we are creating 3 times the growth hormone that we need. Or a mutation could happen where we get a hyperactive or degradation-resistant protein. In addition, we have things that are called tumor suppressor genes that encode proteins that regulate those checkpoints in our cell cycle, right? Tells it between the G2, the S, and the M checkpoints. That tells it to go on. So if they mutate, these proto-oncogenes, there's tumor suppressor genes, can cause the cell to move through the cell cycle without having to stop at the G1, S, G2, M cell checkpoints. 2 examples of these are the RAS gene and the P53 gene. So the RAS gene is a proto-oncogene. Proto-oncogene here, let me just fix this real quick. There we go. So, sorry about that. Whereas is a proto-oncogene. RAS is a proto-oncogene that synthesizes a protein that relays a signal from a cell surface receptor to the cyclin cascade. Remember, the cyclin cascade is responsible for letting the cell continue past the checkpoints in the cell cycle. So normally it requires a certain ligand or a certain signal to bind to the receptor to signal the cell cycle to continue. But if there's a mutation, it might be active all the time and tells cyclins to keep increasing in concentration and go unrestricted through the cell cycle. So here's an example. RAS is this G protein coupled receptor. So when a growth factor hits the receptor, it activates our G protein. That activates the cell kinases and causes a transcription factor to happen, and we go through the cell cycle. A mutation happens where there doesn't have to be any stimulation, but RAS is continually going to activate our cell cycle. Another example is the p53 gene, which is a tumor suppressor gene. And it codes for a protein that's a transcription factor that promotes the synthesis of many cell cycle inhibiting proteins, so those found at the checkpoints. So for example, if DNA is damaged and protein kinases will activate p53, which will inhibit the cell cycle, will cause a transcription of a protein that will inhibit the cell cycle. If there's a mutation in p53, that signal from the damaged DNA won't activate p53, and no inhibitory protein will be created, and that damaged DNA will be replicated and mitosis will continue. So p53 is in charge of inhibiting the cell cycle when things go wrong or to stop it at certain checkpoints, and it activates many other proteins. p53 is extremely well known as a proto-oncogene and is known almost as the guardian angel of the cell cycle. And that's why in almost 50% of cancers, a mutation in at least one form of p53 or another like regulatory P protein, like p11, p43, occurs. Lastly, what we want to talk about is the inherited basis of cancer. So unfortunately, cancer has an inherited basis. The good news is it often takes more than one defective or bad mutation in a proto-oncogene or a tumor suppressor gene to develop cancer. This is why it makes sense that cancer is inherited. Because if you inherit a mutated copy of one of these genes that doesn't do what it's supposed to be doing, and you have you're that much closer to having a total additive effect that results in cancer. So, for example, if you need, let's say hypothetically, 3 different proteins involved in cell cycle regulation have to be mutated, defective to cause cancer, and you inherit 2 of those defects, you're that much closer to having cancer. Examples are, and these are really well known, the BRCA1 and the BRCA2 gene. And they're 2 tumor suppressor genes that are implicated in breast cancer. And the study's been done, and if a woman inherits one mutated copy of either of the genes, so if they're even a heterozygote, having one mutated or defective copy of either of these genes, they have a 60% greater chance of developing cancer than with someone who has 2 wild-type or normal or non-mutated copies of either of those genes. That's just how important those 2 genes are into suppressing cancer, into regulating the cell cycle. That's why it's really important to stay away from mutagens like UV light and chemicals, because some mutations can cause cancer. And if you have inherited defective copies of these proto-oncogenes or tumor suppressor genes, any mutagens are really risky for you. All right? Not to scare you, just saying this is why this research is super important. That's why we talk about this. Sorry to get on my soapbox, but that's why I spend so much time in AP Bio talking about regulation of gene expression in the cell cycle. This stuff can change the world. This stuff right here can change the world and change people's lives. And I want all of you to know about it and to be really knowledgeable about this stuff. Because hopefully you're going to be the people that do that, you know? You're going to be the people that figure out the missing link to maybe turn off or fix p53 to prevent it or make it less susceptible to mutations, or to be able to protect or counteract some possible mutations in the BRCA1 and 2 genes and drastically affect people's lives who might have these cancers. Alright, sorry to get on my soapbox there for a second, but that's it. If you have any questions, just let me know.